Hey biologist, Mr. Fott here. Today we're going to be talking about membrane structure and function. And first up are the must-knows. So why are these membranes considered selectively permeable? Also, talking about the roles of our phospholipids, proteins, and carbohydrates as they relate to our membranes. So first off, we say that the plasma membrane or cell membrane is what's called selectively permeable. And if you break these terms down, to be selective means to be choosy. So not everything gets chosen, only certain things. To permeate means to move through. And so we put the two terms together that only certain things, the selective, can permeate or move through this membrane. So not all substances are allowed to move across more easily than others. Additionally, our cell membrane or plasma membrane is referred to as a fluid mosaic. So, again, breaking our words down, to be fluid means to have like a flow to this. Part of what helps it be fluid, as we'll talk about in just a moment, is that it's going to be held together by these weak interactions. If these were strong interactions, they would be much more rigid and wouldn't have quite as much fluidity as what we currently see with our membrane. Additionally, mosaic. I always like to think of like a mosaic piece of art. And these are the pieces of art that, if you look at the whole picture, it's actually made up of individual little pieces or pictures themselves. And so, just like that, in a similar fashion, their cell membranes are mosaic, and that the big picture, the membrane, is made up of a bunch of little tiny it pictures or images, or in this case, our phospholipids, proteins, carbs, etc., etc. So we take a look at the whole thing, and there's a lot going on here. So we're going to break down a few of these substances, but just know that this right here is more so what in actuality is going on. That we can see here's our membrane in the middle. We have our giant purple blobs, aka our proteins. We have the inside the cell, the outside the cell, and a whole bunch of other things going on. So let's start to break this down. First off, we say that this is considered a bilayer, so that the membrane is not just made up of one layer, it's actually made up of two layers, thus bi meaning two layers. These layers are made up of our phospholipids. As you recall, we have the hydrophilic heads, thus we have the water on both the inside and outside of the cell, and our hydrophobic tail, those are the two squiggly bits that are towards the center here, thus repelled against the water. This is what's referred to as amphipathic, where it is both hydrophilic and hydrophobic. Fun fact that this is actually a little bit where we get soap from. Soap is made from, traditionally anyway, from animal fat, and that is both going to be considered hydrophilic and hydrophobic, which is why it does a good job of rinsing off most things. Additionally, we form what's called a hydrophobic barrier. So notice how we have the water portions are pushed on either side on the outsides there, but our hydrophobic portion is pushed towards the center of this bilayer, which means that things that are of that nature have a difficult time crossing this barrier. So these hydrophilic portions, those heads on either end, are going to actually keep a lot of molecules out because they're hydrophilic, and they're going to push things away that are considered hydrophobic. Part of its fluidity is going to depend on a few things. So, first off, the temperature will actually play a factor into how fluid some of these phospholipids are going to be. So, whether it's going to be a higher temperature or lower temperature, and this in part is going to depend on where the living creature is going to be. So, is it going to be in a desert, in which is much higher temperatures, and they might have the phospholipids are going to be more adapted towards that. Versus if it happens to be a more frigid environment, let's like an Arctic tundra or something, then their phospholipids are going to need to be adapted in a different manner. There are other structures that exist within the membrane itself, such as cholesterol, and part of what that does is it allows it to place in some kinks in there, just so that it can maintain its fluidity. So, like these large bulky structures that kind of push things apart, so they give it some space, just again so that it allows it to then be more fluid in this fashion. There are two types of proteins that we're going to be focusing on. One is referred to as integral, and the other one is peripheral. How I like to think of these two, to integrate means to like insert something in between. So our integral protein is actually embedded like inside of the membrane here. So part of this is that it's what's called a transmembrane. It is going to span the entire distance from inside to outside. These types of proteins, they can then be hydrophilic and hydrophobic, just so they can interact with both portions of our phospholipid bilayer. Additionally, our peripheral protein, this I like to think of if you think of your field of view, your peripherals are on the outsides. So not towards the middle of your field of view, it's on the outsides. Similarly, this is where we see the proteins are either on the inside solely of the cell membrane or the outside solely of the plasma membrane. So they don't exist embedded, so they're not going to be spanning the distance of that membrane. And this is where we're going to see a few specific functions of these peripheral proteins and how they interact with the integral proteins. So these two, they're not like entirely separate from one another. They oftentimes are going to play with each other, as we'll see in a lot of different examples. So here are some images. Again, giant purple blobs that are going to be embedded within our membrane. Those are going to be considered proteins. And we're not going to worry about the specific functions of them and anything like that. Not yet. We'll talk about some examples as we go through. But we can see those giant purple blobs that span the entire distance that are going to go from inside to outside and vice versa. Those are the integral proteins. Our peripheral proteins only exist either on the external side or internal side. So we can see they do not get embedded internally. And part of that has to do with its hydrophilic or hydrophobic properties, in which then it's going to exhibit that allow it to either become embedded or not embedded. So here's a good example of like one of our transmembrane or integral membranes in which it's inserted inside of the membrane. So we can see that it has to have hydrophilic portions, thus we have fluid, either internally, that's the cytoplasm, or externally, that's the extracellular fluid that's on the outside. Both sides are going to have water-like substances, which means it has to be hydrophilic. However, inside, we have those hydrophobic tails, so it must also exhibit those hydrophobic properties. So we can see this is a pretty good diagram just to demonstrate where those properties of those amino acids, be it hydrophilic or hydrophobic, this is where they start to play a factor into how our proteins are going to be functioning, where they're going to exist, whether they're going to be peripheral, whether they're going to be integral, et cetera, et cetera. Now, there's some functions of proteins that are listed right here, and we'll see a slew of different examples. We're not going to get into them just yet, but we can have different things like transportation, which we're going to focus on a lot, enzymatic activities, signal transduction, which is a whole process we'll get into, cell-to-cell recognition, so two cells communicating with one another, intercellular joining, as well as attachment to exo or cytoskeletal and extracellular matrices. One more thing about this membrane that we'll talk about is the function of our carbohydrates. In part, what these are our carbohydrate structures. So notice how they are the hexagon structures. So these are going to be made up of our monosaccharides. Each of those 6 carbon structures are going to then exhibit externally here more so into our membranes. Part of what they're going to do is help cells to recognize. They're mostly like cell recognition. They could also be specific to things like blood transfusions, specific type thing here. We can have glycolipids, glycoproteins. They are typically not going to be just attached to the membrane itself, but more so attached to the protein that's attached to the membrane. So there's a few different functions that the carbohydrates are going to exhibit as well within this whole structure. One final thing, if you're wondering, well, how does the cell membrane maintain its size and shape, and what if it gets damaged and needs to be replaced? So don't forget, we talked about our endomembrane system. So we can see towards the left-hand side, that's the endoplasmic reticulum. Then we can see the squiggles are more towards the middle. That's our Golgi apparatus. And then pushing its vesicle outward so that it can then send molecules or proteins to the extracellular fluid out there. But additionally, that it's those vesicles are going to merge with that outer membrane. Thus, if they need to then insert different proteins or different structures into the membrane, this is one of those mechanisms at which it can then do it. Thank you for listening. Please let me know if you have any additional questions. Feel free to email me at jfod@shoreline.org, or you can send me some Schoology messages. Have a great day.